This video is going to focus on 2 antibiotics that target membrane function and 2 antibiotics that target nucleic acid synthesis. So first up are the inhibitors of membrane function. So remember that membranes are phospholipid bilayers that regulate what enters and exits the cell. And so antibiotics that affect the function of the membrane typically do so by inserting themselves into the membrane and making them leaky, which means that things can leak out of the cell or things can enter the cell that shouldn't be. And this can cause a depolarization of the membrane, which is going to be detrimental to the bacterial cells. So the first type of antibiotics, polymyxins, are going to bind to portions of LPS, which is going to give them access to both the inner and outer membranes of our gram-negative organisms. And it is again going to be able to make them leaky and kill the cell. On the other hand, daptomycin, it is going to be specific for gram-positive organisms. They are not able to get through those porins that gram-negatives have. And so daptomycins don't affect gram-negative organisms, but they are very useful against some of the antibiotic-resistant gram-positive organisms. And this antibiotic works by inserting into the membrane of those gram-positives and causing that depolarization of the membrane, because again, it prevents the cell from being able to regulate what enters and exits the cell. So real quick, here is a picture. This is of polymyxin. So you notice there's that thin layer of peptidoglycan, and the polymyxin is then going to be able to access the inner and outer membranes of gram-negative organisms, making them leaky and eventually resulting in cell death. So the 2 antibiotics that target nucleic acid synthesis have very different mechanisms of action. One is going to target DNA replication and the other is going to target transcription. So the quinolones and fluoroquinolones are going to target a bacterial topoisomerase, which is a key player in DNA replication. And so remember, topoisomerase prevents supercoiling from happening. It relieves that supercoiling, I should say. And so by targeting topoisomerase, that supercoiling will not be relieved, which can lead to breakage of the DNA. Our rifamycin antibiotic is going to bind to RNA polymerase, which was the key player of transcription, also called RNA synthesis. So let's look at pictures that represent these mechanisms of action. And I couldn't find a really great one for our quinolones and fluoroquinolones, but this is just showing that during DNA replication, that replication fork is made. Remember, helicase separates those strands. And by separating the strands, it causes the DNA outside of the replication bubble to become supercoiled. And that's where topoisomerase normally relieves it. And again, these drugs prevent topoisomerase from doing its job, and so that can lead to breaks in the DNA. And remember, for rifamycin, RNA polymerase, look at this nice illustration here, kind of at the bottom middle, showing what should normally be happening, right? Transcription is going to be where RNA polymerase reads the DNA and creates a piece of RNA, right? That some of which is then used to make proteins. And so RNA polymerase is our key player here that's actively making our strand of RNA. And so rifamycin, when it's involved, it can bind to RNA polymerase, which prevents this process of transcription from even happening. Okay? All right, that is the end of this video.